And welcome to Epi Seminar. This is a really exciting Epi Seminar for me. I was really, I don't know why I didn't do this earlier. Someone mentioned like, we should have something on like fecal transplant. And I was like, oh my God. I know just the person. So my old friend and colleague, Dimitri Draconda from VA, as some of you know, before I came here, I worked for 5 years as a research investigator at the Minneapolis Department of Veterans Affairs. So it's 2008 to 2013. Chris was there. And I don't know if this is still the case, but there were these lunch research seminars and journal clubs. And at the time, there was the group was just like bursting with enthusiasm for fecal transplant. And in the lunch seminars, it did not matter what the topic was. It could be on like long bone fractures, and like someone, Dimitri or someone, would be like, "Have you considered fecal transplant?"
Definitely wasn't me.
It was just epic. So like the whole conversation was about like, oh, there's the DEY method, but you should throw away the blender, and like all of this stuff. And I was trying to eat my lunch every week as an innocent epidemiologist. So I had been wondering how your trial had gone, and so now we all get to find out. So let me introduce Dimitri. So Dimitri is the section chief for infectious disease at the Minneapolis VA Medical Center. As an epidemiologist that worked for 5 years in a health system and hospital, I can tell you, from serious Chris, that the infectious disease group, they are the best, like doctors to hang out. They're always the people with the most interesting ideas and you know, just thinking of really like sort of outside of the box school types. So Dimitri has been there for quite some time. He's also on faculty here at the University of Minnesota in our medical school. He conducts clinical trials, as you are about to hear about. In addition to this, he's been in the area of urinary tract infections and optimizing antimicrobial abuse. So without further ado, I'm going to hand it over to you, Dimitri. Thanks for coming and visiting our WBOB. I really appreciate it, especially during this very challenging week that we've all been living through, especially for federal employees.
Yeah, so that was a nice intro. Thanks. Nice to come here. I've driven past the WBOB. I've heard about the WBOB. I didn't know it was so nice. Really wasn't it after long? So I can't even it. So yeah, so as mentioned, it's been a fun week for federal employees. Chris, did you get the reply email?
Well, yeah, I got the, but I'm not doing work with that. You're welcome.
Oh, so you get without compensation.
Yes, but I've been told that everything applies to work it up.
Yeah, so I did not reply, resign, yeah. So I'm still technically at the VA. You know, very tempted, you know, but who knows? So don't have conflicts. There's going to be all sorts of off-label investigative use for seed and seal therapies. And the trial I'll talk about in the middle section is funded by the VA Cooperative Studies Program. I'll just mention what that is because I think it kind of flies under the radar in the sort of research landscape of what it does. I'm going to go through, review the history of microbiome therapies for C. diff, focused again on this VA-funded trial, and also recognize the new microbiome products in the market and the evidence that supports them. I'm going to try to follow my usual rule, which is no give trade names, but it's really hard because there are no non-trade names for these products. We'll get to that later. And then talk about the weird regulatory things that happen around these things, why it's sort of been hard to come up with a FDA-approved drug of something that we don't really exactly know what's in it, and how do you regulate it, how do you put a label on it, how do you tell people what's in it, and how do you charge for it, all those things. So, and then also got to talk about, you know, how the early products are being used. So, so I know this is a mixed audience, so the folks here, like anyone like, oh, C. diff, I know what C. diff is, quick show of hands. People heard of C. diff. So, it wasn't always that way. It was isolated in 1934. I don't know what they were doing, but Hall and Toole were just looking at the poop of neonates, and they found this bacillus, and they said, yep, that's part of normal flora. And then for whatever reason, they said, let's stick it into guinea pigs and rabbits. They're like, oh, it's very lethal to them. Interesting. And, you know, decades went by where we just knew that, okay, there's this difficult to grow organism. It's in neonates, bad for rabbits, so keep your neonates away from your rabbits, I guess, but there really was no clinical implication of it. It was a curiosity that was out there. And then in the late 70s, we were giving people antibiotics, and we saw, not we, the predecessors, I wasn't born yet, but people were giving people antibiotics, and they noticed some people got colitis after antibiotics, and they tried to find what the source was. Usually you couldn't find anything. Sometimes you would find Staph aureus, and it turns out that you just find Staph aureus everywhere. It's just very common bacteria. And so there was this drug, vancomycin, which in the IV form was good for Staph aureus. They knew that, hey, if you give it orally, it doesn't get absorbed, and we're finding Staph aureus in the gut, and they gave it, and it seemed to improve. And so, it turns out that yes, it worked, but they were treating the wrong bug, but it turns out it was treating the right bug. And they eventually found out that no, there is this difficult to grow Clostridium, C. difficile. It's an anaerobe, and then they said, you know what, let's try an anaerobic drug that was metronidazole, that was also worked. And that was sort of the standard. When I went into medicine, it was, here's this disease, you get it after certain antibiotics, you give metronidazole because it's cheap, you could give vancomycin, but it's more expensive. And now we've moved on, we have monoclonal antibodies, this thing called benflutoximab, and we have FMT, which I'll talk about a little bit, which has been used in many places. Usually it's given after someone has improved, like they take medications, their C. diff is better, but they're worried that it's going to come back. There's this high propensity for coming back. And so it's given not to actually treat your episode, it's given to prevent the subsequent recurrence that can happen. And so that's what we'll chat about a bit more. So before we're going to talk about that, just briefly, C. difficile, if it comes back, it's bad. So it can be debilitating. People are terrified of going out. You get admitted to the hospital, you have lots of clinic visits, you have fecal incontinence, you're on antibiotics, you lose weight, your quality of life really is terrible. And I would have to squint in there. I think they're saying, 12,800 deaths, $18 billion from the CDC's 2019 antimicrobial threat report. So it's a big deal. I think you can see, so here's how we currently treat it. So there's the most recent updated Infectious Disease Society of America guidelines. Basically, they say that for the first presentation, you either give fidaxomicin, which is mildly controversial. It's the most expensive antibiotic that exists, about $3,000 for a 10-day course. Alternatively, slightly less effective is vancomycin, which costs about $20. So you have to decide whether you want that extra 10% reduction in recurrence for $3,000, or do you want the $20 one and have a little bit higher risk of recurrence. But when you go down to the second or subsequent C. diff recurrence, the one that they suggest all sorts of things like a taper, eat a drug, and then fecal microbiota transplantation is tossed in there. So what the heck is that? There's always these references, and I can't read Chinese, nor would I be able to find it, but supposedly there's in Chinese writings from 1700 years ago, broth with dried stool from a donor. You mix it, you give it to patients with diarrhea, and they get better. I cannot confirm this, but it's referenced in every single C. diff talk on FMT, so presumably it's out there. It turns out veterinarians do this. So that is a fistulized cow where they actually put a fistula into I'm not sure which of the four stomachs you access there or the rumen, but you do it so often that you just create this opening and put a cork in it. So whenever you need to get it, you pop the cork out and use that device and suck up some of the good fluid and spray it on the food of your sick cow, and the sick cow eats it and they get better. So that is transplantation, and that is for cattle ruminal acidosis. Don't know the mechanism, but it gets better when you spray healthy cattle rumen fluid, which is, on the way to becoming fecal material. So when did we do it in humans? So go VA. So this was described in 58. VA surgeons do things just because they feel like they can. For patients, and again, they did not know this was C. diff. They just had diarrhea. And the donor was a normal subject who had received no antibiotics during the previous several months. Rumor has it that this has been passed down by lore. George Sorosi tells me that he confirmed this, that it was a medical student who was just told to go to the bathroom, come back with a sample. So this is what you used to do with a med student. So yeah, it's not clear this was C. diff. It was that people were having diarrhea post antibiotics. There clearly was no IRB or consent. There was no standardization or screening other than you look pretty healthy as a med student. He was told to never do this again, but he did publish it. So that is where the world FMT literature started. So if you search fecal transplant, it starts at 1958 and Neisman, and it kind of trickled around. You can't really see, but there's like 1-2 papers a year, and then just for reference, that's 2006, 8. And last year, it jumped up to 1,300 papers a year. So it has just exploded in interest. So everyone likes FMT. Now, should they like it? Who knows? But everybody does. And why? So this is, again, the CDC's antimicrobial resistance threat report. This is what every grant person who's writing about C. diff starts off with. Like, we get top billing at the top 5 with carbapenem-resistant Acinetobacter, Candida auris, all these really bad things, C. difficile, which is a little bit weird because it's not terribly resistant at all. Like, so this is the antimicrobial resistance threats. And I guess the concern is that it just keeps coming back and you keep getting antibiotics and it will drive resistance to other things, because there's actually almost zero resistance for C. difficile itself. So, basically the state of the field from 2000 up till a couple years ago, just a ton of case series, almost all uncontrolled. And these were the things that Rachel was saying, oh, FMT works for everything. It's perfect. It's magical. Success rates are up greater than 90%. There were some small RCTs. The first one got in the New England Journal, and like, you do the smallest trial, you can get in there. There were some issues with it, which I won't go into two things, but basically there were a ton of different methods. Do you give fecal material via enema, via colonoscopy, via NG tube, via capsules? How do you do this? And so again, this was the one in the New England Journal. This was sort of launched it. The weird thing was, so they again claimed 81% success, so a little bit lower than the case series, which is usually the trend of, like, when something new comes up, the case series say it's perfect, and you start doing better study. Like, yeah, it's maybe not perfect, but it's better. But note that the control groups were just terrible, like 31% to 23% success, which is, if your control group is that terrible, almost anything looks good, and that is not what we see in clinical practice. You do not expect to see 70% of recurrence rate. So it was a bit weird. So we weren't really truly convinced. And so people were like, if this were a drug, you wouldn't get approval based on case series. You wouldn't get approval based on controlled trials. You wouldn't need to do an RCT with a placebo. What drugs need to do to get to market. And so a few people did this. So in Canada, Susie Hota did this study, fecal transplant versus placebo, stopped for futility. Also a little bit issue with enrollment, because there was so much press about it that it was considered so great that no one wanted to enroll in a trial where you only had a 50-50 shot of getting it. So A, futile, B, they were having very significant enrollment issues. With Kelly, this was an interesting trial. It was donor stool versus your own stool. So everyone was told go to the bathroom, and then half the people that threw it away, and half the people that mixed it up and gave it back to them via colonoscopy. And it worked. I mean, they got a publication in Annals. They showed the recurrence decreased. But it was weird because it worked at one of the two sites, which sort of raises questions like, maybe it's not the poop you're giving, maybe it is something that they're doing at one of these sites. So one of them was very popular. It was a long time to get in, and you had to be on sort of suppressive vancomycin therapy for months and months and months. And the thought was that maybe it was the long months of therapy that actually did it, and not the stool. And then what I'm going to talk about here, so the RVA trial, FMT versus placebo, and I guess I gave away the ending. It stopped for futility, but more interesting than that. So has anyone heard of the VA co-op study program? Chris said, "Don't you have?" They've had. All right, well, good. This is good. So I think it's weird. It's a sort of hidden gem of medical research. You can read about it at that website. They started off in 1946, 10,000 veterans with tuberculosis, like, does streptomycin work? Dr. Barnwell was the leader of that trial. That's now the biggest award you can get in the VA for clinical research, and a couple of folks at that Minneapolis VA have got it, so kind of neat local connection. And the structure is, there's, in everything in the VA, there's central office, which is located in DC. Still, I think, who knows, they've all resigned. And there's coordinating centers where basically there's a whole bunch of experts in clinical trials, biostatisticians, data people, all sorts of people who run trials for a living. They're located at these various sites, and there's epicenters where they do the same thing, but they do epidemiological studies. And then there's this big facility in Albuquerque, the Clinical Research Pharmacy Coordinating Center, where it's this big state-of-the-art kind of manufacture placebos, drugs, in CGMP manufacturing conditions, and they take an enormous part of your budget, but they do good work. And they also take your samples and freeze them and all sorts of other use and stuff. So what do they do? Central office just kind of provides oversight. What are we going to fund? What are we going to prioritize? sure they are fun time these days. The coordinating centers have trial expertise. They lead they have data source, statistics, analysis, implementation, all the nuts and bolts. They're kind of stuck in their ways. Like when you ask them, why do we do this? Because this is the way we do things. And well, we might have a better way, boys. That's a good. The coordinating center does all that stuff I told you about, and then there's these network of dedicated enrollment sites that are basically sort of hospitals that do a lot of trial and have expertise at starting up things. How do you train a new coordinator? How do you get them to fill out paperwork so that they don't fail their first audit? How do they stay out of trouble with the IRB, et cetera? And then at the individual VA medical centers, you enroll patients, the study coordinators assign to enroll patients, and there's some local site investigator that gets protected time to sort of do the oversight and answer questions of the study coordinator. So this is sort of a very simplified version of what happens. It's actually the most simple funding mechanism I'm aware of. The initial LOI is 10 pages. It's not terribly structured. It's just like, tell us your idea and see what happens. And 10 pages is a ridiculously low entry to grant funding. And so you're like, yep, this is what I want to do. And you send it in, and they sort of look at it for administrative review. Like, does this pass the smell test? Does this seem like a decent idea? Yeah, it does seem like a decent idea. They will then send it to external review, looking for topics, subject matters in the field, and ask like, hey, someone sent this in. What do you guys think? They will send it back, and if the scores are good, you get approved or disapproved for planning, and they invite you all to DC. Maybe now it's virtual. But at the time you go to a planning committee meeting and they bring a bunch of experts that you've never met, you bring along a few people that you think will be helpful to you, hopefully people who are favorably inclined towards your study, who have a gravitas in the field to sort of say, yes, this is a good idea. Maybe not too much gravitas, so they don't take all the thunder in the room, so there's a little bit of a balance in that. So the soft skills of this, and then you show up, and then you come with an idea. Our idea changed quite a bit because a lot of people in there are pretty smart. Like, I don't think you should do it this way. You should do something else. And so you tweak it. And at the end, you present to the research leadership of here's our current idea. They decide to go on. The second planning meeting is almost a year later. In between, you write your protocol. And then the big white box is just called the Co-op Studies Scientific Evaluation Committee. It's really stressful. It's basically kind of an oral defense of your project. You show up. We'll cover that later. But what we did, so this was in the blue box there, the planning committee. We came up with this idea. So we decided that we would do this. We had to come up with an acronym. This is not the best acronym, but it was not the worst acronym. Match. So and the at home is going to be. We'll show you why we did this. So we were going to say, all right, if we really want to get this to work, it's got to be randomized, it's got to be blinded. People can't know what they got because there's so much belief that this thing is good. And you might say, well, who cares if it's a pretty hard outcome. It turns out diarrhea is not the hardest of outcomes. So not to get too gross, when people are stressed, and everyone who has run a marathon or done any of that knows, the reason there's 100 porta potties out there is everyone's stressed out and everyone is having change in their bowel movements. Most of which would qualify for yes, you meet criteria for testing for diarrhea based on what's going on this morning. So we said that really has to be blinded. We made these assumptions about recurrence, that there'd be 35% placebo, and we'd look for a 15% reduction, so down to 20%. And we said we need 390. We later tweaked that by saying, okay, we'll go to 85% power instead of 90, and we will lose fewer people, and tweak it down to 318. So this was what we came up with. We would take patients who have recurrent C. diff, we would centrally identify them, contact them, get their baseline info. They would finish a course of standard antibiotics. Everyone who didn't improve, you couldn't enroll, but people who improved or resolved, they would get their time off of antibiotics. So that you give them their fecal transplant, there were still antibiotics in their gut to kill the new bugs you're giving them. And you randomize them to placebo capsule or FFT capsule, and follow them for 8 weeks for primary, which is kind of the standard field, and also for 6 months for safety. So this is the thing that all the letter writers to our trial fixated on. The primary was recurrent C. difficile, definite or probable, and death within 56 days. And most trials do definite, so that's people with diarrhea and who have a stool test for C. diff. VA was very patient. They were like, no, we want this. Patients do not care if they have diarrhea that is negative for C. diff or diarrhea that's positive. All they know is they're having diarrhea and incontinence. This has to work to prevent it. We don't care about, so count it if it is diarrhea, even if there's not a positive C. diff test. So we did that. That's why you're probable if you have diarrhea but no test. You're definite if you have diarrhea plus a toxin test. These secondary outcomes are there, and then we're exploratory things. We're going to look at the fecal microbiome in the patients pre-transplant and after transplant, and does it match up with what we gave them. Then looking at adverse events and serious adverse events. So this is the crew after the first planning committee meeting, just to highlight in the red sweater there, Osma Shaukat, who's my co-chair, so we did this all together. The guy in the yellow shirt is the guy who is in charge of the co-op study program, whether or not you get money or not. So he gets to be in the picture. So there was this unique wrinkle in ours in that they had already funded the year before a trial for C. diff, looking at trying to prevent recurrence. So that was a big deal. And they had already said, yep, we're going to try to prevent it with either this new drug, the $3,000, of course, one called pedaxamycin, compared to an old one, given either a standard dose or a tapered dose, and it had already been approved. And that having two CSP trials on the same disease, it's not unheard of. There's no rule against it, but sure makes life difficult. The standard study is like, okay, here's the 30 centers with the most cases, park a study coordinator, each of those, recruit from those sites. And so we're like, right now, the 25 to 30 best centers are off the table, including Minneapolis, but we said no because we kind of thought this was coming. So we were still on the table. So we came up with this idea of that, okay, we're not going to do 25, 30 local sites. We'll just have one site, and we will recruit from any VA patient who has recurrent C. diff. And we got time because they get this, at least 10 days, often longer than that, plus a washout. So you have time to contact them, first identify them just electronically, see if they're eligible, contact them, see if they're interested, and then book travel, fly a coordinator with their assigned bottle of randomly assigned treatment, which we had to can you go through TSA with Philip? Can you what are the rules on this? Yes, you can. So it was kind of interesting. They would go out there and they have a one-time administration of capsules, and then the rest is all phone follow-up with mailed stool samples if you had recurrent symptoms. So 11 months later, you go to this CSEC review, which isn't the stressful one. So you get the reviews back. You're supposed to get them a week before someone was delinquent. We got them, you get it the night before. And basically, it's an oral session. There's 10 members, and they just list off all the things. In our case, it was 17. And you could tell, yep, these are all from the reviews. And you get 30 minutes to orally address, what are you going to do to bring these? And then you leave, and they talk, and it's weird. They're in a classroom, so you can kind of see what they're doing. And you're just trying are they smiling? Are they shaking their heads? Are they laughing? And then they bring you back in, so there's no waiting from them. They just give you a thumbs up or thumbs down. And I guess there's more than just thumbs up or thumbs down. Thumbs up is, yep, you're funded. Partial thumbs up is you're funded for a pilot study. You show us that you can do it at a limited scale, or come back again, there's serious issues, or don't forget it, there's really serious issues, you're done. So we got a thumbs up, and we're very excited. I never wear ties or sport coats. Clearly, this was a big deal. So, this was, conducting a decentralized trial was kind of unique. Everything happened at one VA, which was beautiful from an IRB standpoint, because there's been one IRB involved and not many others. Lots of meetings, and then the real work is the study coordinators, the programmer, and the site investigator. So we had a really outstanding programmer, sadly passed away since then, who basically developed this online search where he pulled out all sorts of things that would suggest that there would be a case of recurrent C. difficile from the VA corporate data warehouse, which is this big data set that gets updated daily. Team would review them. Anything that looked to meet inclusion criteria, we would reach out to them and say, can we contact them? If they said yes, you can contact them right away. If not, you would send a letter, and you have to wait a week until they did not opt out. Then we'd ask them participation, if they're interested. Then we schedule travel. We have this very quick, usually VA travel is a nightmare. It takes a very long time. So we had to get pre-approval that yes, we're good for the money. If there is a urgent need to book a flight, just do it. And they did, which was good. And so if they're interested, they would travel a patient with this randomly assigned bottle of pill that could consent them, enroll them. And sometimes it was at home. Some veterans did not like people coming to their home and wanted to meet at Denny's. So if you see people doing interesting things at a restaurant, that's what's going on. They would, sit there and take their hoop pills know, so, wipe down those tables. And the follow-up is via phone, record review, and then stool samples to the central lab. So again, the pros of this were everything is at one site, one core team of study coordinators who really knew the study, really knew the inclusion and exclusion criteria. If they had any questions, they would just shoot us a message. They could text us like, hey, got a quick question for our eligibility. So really easy to troubleshoot things, change approach, etc. And the cost savings is pretty substantial. So if you go the traditional way, you have a study coordinator parked at 25 sites. Each of those is roughly 100,000 in terms of salary benefits. So you're looking at 2.5 million annual salary. We were going with 2 to 3 coordinators and a $500,000 travel budget, which seemed excessive, but then like, wow, that's still quite a bit cheaper than the other way around. So we're fine with this. The challenges are that there's no local presence at sites to raise the profile or to help with enrollment. We were like sending out emails to the VA ID and GI listservs, like, hey, there's the study, happy to enroll your patients. But some people just have never heard about this, and they're like, wait, who's contacting you, and why are they offering this? Can they do this? So they really didn't know the study team. We would try to reach out. They had materials like, if you have questions, if your clinician has questions, they can reach out to us and see what's going on. There was an issue with Pennsylvania. The Pennsylvania Supreme Court says if you're doing a procedure, the physician must consent them, not a member of their team. So if we had to get through an IRB hurdle where one of us actually had to do the informed consent for it, we would have logistics of travel. You would end up and get a call from our study coordinator, like, yeah, I'm at the patient's house and no one's answering the door, and they would just get ghosted and travel to North Carolina for good reason. There was safety concerns. Everyone at first was like, you're going to send the study coordinator out to a house, they're going to get kidnapped and dismembered. What are you doing? And so we had to okay, well, we send people to people's homes and healthcare. How do we currently do this? What's the protocol? Let's adopt that. A safety check. Are there any warning flags in the record? If there are, we go with 2 people, etc. And there are all sorts of stool testing issues. Everyone says, yes, absolutely, I will send the stool sample without diarrhea. People lie, they do not send the stool sample without diarrhea. And then COVID came in the middle of all this. And obviously the bad thing was, COVID. Everything stopped. And obviously getting on planes and traveling to veterans became a no-go. In fact, one of our study coordinators, we thought they were going to be our first case of COVID. It was just flu. I remember swabbing them out in the parking lot. And there was questions like, what if COVID is a stool? What if we're giving people COVID via fecal transplant? And so everything was stopped. And then you could be, tests were so unbelievably scarce that you couldn't just, the thought of using the screen asymptomatic donors was not really something that could go anywhere. But actually there were some pros to it too. It turns out we had a bunch of stool from beforehand. And so that was okay. And then eventually we could test donors. And we also realized that, a lot of what we're doing is virtual. Why not all of it? So instead of traveling, how about we just do a bunch of we'll ship the consent form, we'll watch the signature via video visit, we'll ship the drug, watch it be ingested. All of this can be done without travel. And it made us actually feel kind of stupid that, we really did not need to go anywhere. So we decided, let's try that. And the downside of that is that some people just don't have good internet coverage. So people just couldn't do the visits. Some people just, you're trying to do a video visit with an 85-year-old veteran. It does not always work. My mother could never do this. But despite all of that, people really preferred it. So once we offered them both in-person and video visits, almost everybody opted for the video visits. They thought that was much more convenient. So everything was going great. We had okay, we overcame COVID. We're enrolling slower than planned, but we got an extension. I was on vacation and we got a call that we needed an urgent meeting after the Data Safety Monitoring Committee. And this was a scheduled meeting where we had 50% of the planned subjects enrolled. And that's okay. Well, it's 3 things that are happening. It's that either FMT is so good that it's not ethical to continue, or it's just not doing anything, or worse, it's unethical to continue, or there's just, so little difference that it's futile to continue. And so, that's what we had. It was so little difference in the outcomes that it was futile to continue. So we had reached 150. So at 151 of them had reached their 56-day follow-up. We had a little bit more primary events than we had expected, but at this point we didn't know what it was, treatment A, treatment B. So basically the primary endpoint was met at 32.9% treatment A and 29.3% treatment B. There was clearly no difference. The statisticians in the room will know this better than I, but they basically look for the conditional power, like what's the chances that you will show a difference at full enrollment? And using our hypothesized event rate, there was about a 16% chance that we would show a difference, but using the actual event rate we were seeing, it was less than 4%, and that met our criteria for stopping. So the Data Safety Monitoring Committee made that recommendation, and the sponsor said, yep, we're taking that, and everything came to a halt. Actually, our last study coordinator was on an airplane when we found this out, like to the point where it was like, should I get off the plane? Because like the door is like about to close, they couldn't get off. Like, yes, get off. So we saved a little bit of money, very efficient. This is ultimately what the results are, and so you'll see in panel A is the primary endpoint. So overall, it turns out that the dark is FMT. So this is, did you have definite recurrence, probable recurrence, or death? 32.9% versus 29.9%. That second one is stratified by, is it one recurrence or more than one recurrence? So have you had, just your first episode of recurrent C. diff or multiple episodes? Maybe a hint that with more than one recurrence, so okay, 37.5% recurred versus 50% with placebo, that was not significant. And if you go by possible, similar. If you go by definite, clearly there were less definite. If you go by death, there was really nothing there. So nothing that significance. And this kind of, yeah.
Sorry, is that in those who had previously had more than one recurrence, a different population, or that's different out?
That is a different population. So you could enroll in the study if you were having your first recurrent episode. So you had C. difficile once, and then your second episode is your first recurrence, and you could then get FMT or placebo. Some people had more than one recurrence, which is the guidelines suggest it can be given for both first recurrence or the second or fourth recurrence, but probably the patients more at risk are those who had multiple recurrences. They've already shown a tendency towards recurrence, and so if you really wanted, there's some thought that maybe we should have limited it to just those having greater than one recurrence. So in hindsight, would we like to focus on those? Yes. Also, it's very hard to find those because it turns out that a lot of people who say they have multiple recurrences, they have a lot of GI issues. It's not exactly clear, do they have true recurrent C. diff or are they having motility issues after having a lot of infections? Lots of antibiotics, and there's post-infectious irritable bowel issues, so it's really hard to tease out. So basically, this landed in it had gone from, this was so popular, like I can't remember for it. I think this was pulled from this New Yorker article, which if you're interested in this, it is a bit overzealous if you ask me, but it is a very entertaining read. It has a lot of the people that we recognize, like the company they talk about. We tried to contract them for a year before they kind of were lousy with us, and we said no thanks, but it's got the great name of the experiment. And from there, it's the closest thing to a miracle I've seen in medicine, which you always got to be leery. This is one of the investigators, and this plays when you're describing your things as a miracle, you're a little bit questionable. So it's, everyone thinks it's highly effective, but this is now, there's 3 true placebo-controlled trials, one of which is positive. That was the one where it worked in one site but not the other, and 2 have been stopped for futility. So I think that's a little bit of an open question. Is it really the closest thing to a miracle in medicine here? Now, there is all sorts of variability here because some people, we got our source from colleagues at the University of Minnesota. So Mr. Woman's tried to call me. There's other places. Everyone knows about blood banks. There are stool banks. OpenBiome features heavily in this article. They've since kind of shut down because of all sorts of issues, but this was a place that they basically had healthy donors depositing stool, and they mailed out thousands of these and did relatively scant data collection of what happened with them. But they would say, oh, we've done it thousands of times, we haven't heard much bad, so it must be working. And people, well, you can't really just rely on you haven't heard anything bad. You really do need to ask, follow the historical slate. And then there's a whole regulatory, I skipped the DIY, was tossed in the title, but didn't want to cover too much, but really, you can go to YouTube and type in "how do I give myself a fecal transplant," and you will find various recipes. That, and there's also I think there's an academic paper out there like giving you the list of things to buy from a drugstore, and how you can do this at home. So this has been done. And it really is very murky in terms of what to do for regulation. So the Food and Drug Administration, in May 2013, they said this is a drug, and if you are doing it, you need to. It's not an approved drug, so you need an investigational new drug license to be getting this. And this created a huge firestorm. All the people that were thinking this is the best thing ever said you were withholding this amazing treatment from patients. You're killing people. So the FDA backed down and they said, okay, we still think a drug, but we will use enforcement discretion. So you can give it to people for treatment of individual cases. You have to get informed consent, written informed consent. If you're doing research, you have to get IND, which we have to do. And they did update this, which I'll get to later on. So, of course, at some point with all of this, we're like, we really need to have a standardized product. So there are now, no one knows what to call these, commercial FMT, FDA-approved FMT. I've actually got with FMT-derived commercial products. And the rationale for this is that, you can't just rely on people getting picked from wherever, from some lab, like who knows, like presumably there are differences. And you really need to have a standardized uniform product as best you can, because we really can't characterize what's in there where, we'll get to that. And, you do have to have high quality data that the FDA needs for approval. And if it is effective, this would improve access. Because currently, if you are close to a site that has an investigator doing this sort of thing, you can get FMT. But if you are not close to such a site, you're out of luck. So people travel from long ways away to come to places like the university, Cleveland Clinic, places that have a FMT program, but it's really not widely available. And the thought was, if you had a FDA-approved product, that it could just be obtained anywhere. So there's two of them out there, and do they work? So this is where I wish I can't really use trade name, because all I know is that it had some weird letters and numbers, but it's called Rebiota. It's actually made out of Roseville, Minnesota. I guess I used to be their study data monitor like a decade ago, so I don't have any conflict there. So this is a rectal microbiota suspension from human fecal matter obtained from qualified donors. My son got a mailer asking him to donate, so I don't know how qualified they are. He was very intrigued by that. They would offer, I can't remember what it was, but if you lived close, you had to go in person to the center in Roseville. You could donate twice a day, 7 days a week. And if you maxed that out, it was not bad. But so again, I'm not sure how qualified these donors are. So, and what's in it, we don't know. Like, it contains between 1 times 10 to the 8th and 5 times 10 to the 10th. Like, it's a little bit weird. We don't give aspirin in there. Well, this contains, 300 milligrams or 30,000 milligrams, something in there. Okay, so it's a little bit odd, including 1 times 10 to the 5th of Bacteroides. There's nothing special about Bacteroides. They just pick that one. So you get a one-time enema 24 to 72 hours after antibiotics. And it was a really weird approval. They were having trouble enrolling, and so they, normal approval is 2 randomized placebo-controlled pivotal phase 3 trials. That's a standard that you need to get a drug approved. They basically said, we're having trouble enrolling because there's so much FMT out there, so many people are using it, and we can't enroll patients. So the FDA said, all right, we recognize that you're having difficulties, so you can do 1 pivotal trial, and you can borrow some patients from your phase 2 trial and mix them together and do some fancy stat stuff in there. So basically, 1 phase 3, patients came from a phase 2 trial. This is kind of messy, so I don't know how to basically they pooled it all together, and in the end they said Rebiota, that's red, 70% success, placebo 57, which again, remember, they were saying that early on, it was like, this is greater than 90% effective. And here you're looking at like, okay, it's 13% difference. In this one trial, this was group A was 2 doses. So this is the first study. So group A got 2 doses, group B got 2 doses of placebo, group C got 1 dose of Rebiota, 1 dose of placebo, kind of a dose finding thing. And frankly, 2 doses of Rebiotics versus placebo wasn't significantly different. 1 dose of the fecal transplant versus placebo wasn't significant. If you pool them all together and compare both of them to placebo, you got a 0.047, so just barely meeting significance. In the end, when they pooled everything, they said, okay, the treatment effect is 13.1%. That's the 70.6 minus 57.5. But at the confidence interval, it's as bad as 2.3% better or as good as 24% better. And 2.3% is not 90%, which is what everyone was saying before. I mean, this is not terribly impressive. So they basically said, well, it's 99% certain that it's better than placebo, but how much better, who knows? And they got it approved by a 13-4 vote, so not unanimous. Obviously, this is no fun. Enema is nobody likes to do. So other folks went to something else. This is called Faust. Why Faust? I don't know. SEER 109, this used to be series. So this was, again, human material from qualified donors, free for pathogens. They treated it by alcohol. And so basically you're getting just spores. And then they filter out stuff and they say that, okay, well, again, there's this, tenfold difference of firmicute spores, 4 capsules daily. They did it for 3 days after successful antibiotic treatment. And you had to take purge first. So you did magnesium citrate or polyethylene glycol to sort of empty your bowels beforehand. Now they had more.
Are they swallowing the tablet, or are they just?
They are swallowing these. Yeah, they are swallowing these things. So, and they got much. These are kind of similar things. So panel A is recurrence. Panel B is just the mathematical reverse. So if you didn't recur, you have a sustained clinical response. So depending if you like high numbers, you have 88% sustained clinical response with series 109. If you like low numbers, you only have 12% recurrence. Versus 40% or 60% depending. And so that was a much more robust looking graph. Like, okay, 12% recurrence versus 40%, that's more legit.
Yeah. Would you say the timing of this relative to the cessation of?
They did 2 to 4 days, I believe. Did I put that in there? 2 to 4 days after successful antibiotic, yeah. And there's all sorts of debate about should it be sooner or later, etc. So here's the difference. So the rebiotic enema times 1, they say 71%. These capsules times 3 days, 88%. Notably, in the phase 2 trial, they almost failed. When they did it, they only gave capsules once, and it was no better than placebo, and they recalibrated and said, let's give it 3 days in a row. So maybe that's what you need. You just need more of it. And so maybe if you give 3 enemas in a row, or maybe if we would have given pills 3 days in a row, maybe all that would have worked. Because all the other trials, our trial, the match, and these other 2 that were either mildly positive or also stopped for futility were all single dose interventions. So perhaps we just needed to give more of it. So maybe it's, higher dose is better. 71% and 88% are not hugely different in terms of Faust and Rebiota. Maybe that's just chance. These are, for IV trial, these are pretty big, 182 and 267 people, large for infectious disease studies, but, tiny in terms of cardiology and other things. So funny things could happen with small numbers. And then there's other stuff coming out there which seems sort of promising that higher dose is, I don't even know which company makes this, but it's currently designated VE303, where high dose versus low dose versus placebo, you see recurrence in the high dose was 13%, low dose and placebo were 37 and 45. So maybe it is, you just need more stuff going down. So one last little thing, I'm almost at the end here, so that just regulation, the FDA and FMT, this long, messy relationship. With most drugs, we know exactly what's in there. We know the active ingredient, we know the filler, we know that there's gelatin or talc or whatever. Nobody knows how to characterize stool. And even if you had a donor who's like you completely characterize, they go out and eat some novel meal, that changes their microbiome. Is that a significant change? Who knows? So a lot of people thought that the only ones that are going to get FDA approval are something that's some degree of standardization. It is standardized, but you still cannot go to each of them and look up, tell me what's in here. All they can say, well, it has these things, in this range, but we don't know what else is in there. There may be fungal residual organisms. There shouldn't be viruses, but there might be. I mean, there's a lot of stool. There's tons of viruses in your stool, as we know from wastewater surveillance. That's where we are getting most of those things for COVID and flu. That's I figured that out. So basically, the FDA said, all right, we're going to keep going with this enforcement discretion that you can give FMT without an IND. the stool banks are out. You can't have a stool bank. We think the risk is just too high. If you're having centralized manufacturing, you ship it out to thousands of people, we just don't like this. You're going to need to get an FDA approval. So basically, they need to have an FDA-approved product or operate under an IND, which means they're kind of going out of business. And then when to use or uptake and cost. So the Infectious Disease Society of America says that fecal transplant should be used for the second or later recurrence. They're approved for the first recurrence. I'm not sure how much Biotech is being used. I actually talked to the drug rep, which that always makes me uncomfortable, and she was not able to tell me. Faust is interesting because Series is its Series Therapeutics. This is their only product, so you can kind of track their stock price to see how their only product is doing. And, when they came on the market back in 2015, well before any data came out, everyone thought this was great. FMT is the way for the future, fabulous, $42 a share. I can tell you they got approved in 2021. There's no real movement there. They've only gotten worse since then. So it has not been a commercial success. They're doing what the media is doing, strategic restructuring. Not only doing it in a more systematic way than sending out emails saying, reply with resign if you, but they're expensive. So Vow's, the pills are at 17,000. Biotech for the enema is 9,000. I have no idea how commercial insurers are doing it. I suspect they're doing all sorts of stuff like, no, we're not going to approve this until you fail multiple treatments. The VA, we just sort of made up criteria like, okay, you have to fail one of the standard drugs. You have to try this IV monoclonal antibody, which is pretty standardized and relative to these cheaper. And afterwards, if you still have recurrence, we will approve it. We have not given any as far as I know at the Minneapolis VA, but we're allowed to. That's there if we need it. So basically, this is the end of the story. So it's gone from this 58 case report of a surgeon grabbing a med student. Now we have multiple placebo-controlled trials. And those trials really haven't been terribly impressive if it's a sort of non-commercial FMT. The FDA-approved ones, have better efficacy. I would say the oral ones seem to have the best, 20 versus 40% recurrence versus 12%. And maybe it's because of the 3 days dosing. That's the one that if I had to pick, I would go with. But I'm not sure, like long term, how much is it going to work? Whether payers will approve it, we really don't know. And until there's better data supporting the sort of stuff that comes from a lab, it's difficult to recommend. But I also will say that the field has not been stopped by this. There are so many people interested in fecal transplants. So the diseases that are being studied, are not just C. diff. C. diff, I can at least sort of see the rationale for, like, okay, you had normal bowel flora, something happened, it was returned to normal. People are looking at it for autism, diabetes, metabolic disorder, cancer, epilepsy, everything under the sun. People are doing trials of FMT. If you go to clinicaltrials.gov and search fecal transplant, you will find just page after page after page of study. It's kind of remarkable. Everyone thinks the microbiome is the greatest thing ever. Now, it probably, yes, they all see these statistical signals that, in this patients, they have a different microbiome than normal. And that may very well be true, but whether you can change that by giving them some poop, I'm not sure you can. I have a tough time thinking that, okay, someone who has, metabolic syndrome and is, have obese with diabetes, that happened over many, many, many years. Can we just say, here, take some poop pills and everything will go away? I'm very, very skeptical on that. So, anyways, that is all I got. I will love to take any questions. I'm sorry for the beeping. Apparently, there's a lot going on. Oh, one last thing. I do want to quick say, so, Carolyn has a really fast trials that are quick. This is not where to go. The CSP program is not fast. When I first had this idea and submitted the LOI in 2011, the kids were drinking out of sippy cups. And at publication time, at least one of them is drinking beer. So, fast sleeper.
Thank you so much, Dimitri. That was fantastic. I need to ask you, what did your 4 valve pills look and taste like
I personal experience cannot tell you. They looked like a double O encapsulated product with a acid resistant. We actually had a picture of it in our consent form, so they could look at it next to a dime for size comparison. So they were double encapsulated with acid resistance, so they were very specific. We're told not to chew these. Thought that was obvious, but when they had to put it in the consent form, so it looked like just a rather large capsule. That double O if you'll look at it for size.
Was it like the clear kind of capsule, or was it?
They were not clear, and they were in this. When we shipped it, they were, they had that, they could have dissected that, they could have opened them, they could have done whatever the heck they wanted to them. But when the coordinators were going there, they watched them swallow them, so they did not take them apart. Could they have done so later? I guess they could have.
Like, questions, Abby. Really nice talk. Thank you very much. Full disclosure, I was a failed donor for a certain academic process.
I still speak you up for.
Kept getting kicked out and my kids getting sick.
They like to claim that, oh, it's harder to be a donor than it is to get into Harvard. And then when my kid got the line, I'm like, really? I don't know. Like, he was out on Harvard.
A little bit unscrewed. It was definitely a stringy process here. I would ask you, now you've gone through the process, you've been paying attention to the literature, what do you think is the optimal timing for administration? Like, if theoretically you could actually change somebody's gut microbiome ecology, when relative to antibiotics, like, is there an optimal window for when things have been disrupted and you could actually get new engraftment of new species and get a better situation happening?
The letters to the editor have really focused on this. They were like, you did it too late. Like, there's some you passed a magic window, which they hypothesize exists, and we pointed out, well, no one has defined the magic window, so it's not like, it'd be nice if there was a window, then we would certainly try to make it, but no one knows. No one has any data saying that, hey, if you give it at day 8 versus day 4, you have decreased your chances by X amount. There's unknown. Well, we strove to do it early, not too early. Like, you will find, like, so people have looked in the stool for residual antibiotic levels, and you definitely find it there, and you find it 2 to 3 days out, and that probably depends on motility. Some people have faster transit times than others. So it's probably in the 2 to 3 days, somewhere in there. We, in response to, like, we had this data, so we looked and we said, okay, those who got it within 4 days versus those that were greater than 4 days, was there any signal towards change? There was not. So, what else?
I'm just curious with the FDA trials of some of the revival that.
Yes.
Did they have definitive recurrence as their endpoint? Because, as you were saying, there's diarrhea and diarrhea. Right? And I would hope that they had,
They started off with this gets actually a lot more complicated because it depends which C. diff test are you using, because they went with, the early ones were using PCR testing, which frankly, if we had done that, almost anyone would have it because PCR testing persists in the stool for a long, long time. And eventually people pointed out that, look, if you're doing PCR testing, you might as well go with just diarrhea, because anyone who's had C. diff and gets diarrhea, you will still find a positive PCR test. So the current approach to C. and if we were applying now, I think the field would to do this, you really need to show the toxin is present. So you'd want to do a direct antibody test to see, can it detect the preformed toxin in your stool? So I believe the latest 1,000 use that, but all the Biotech, the first those trials used PCR testing, and yes, they need a test plus symptoms.
And then second, are you done with the poop pills, or?
I'm pretty done with the poop pills. It just learned a lot. It was a long process. It turns out there's a lot of very strong feelings in the field about this working, and it's just really, some of it is just a little too murky for my taste. You had a question there too. Sorry, no, you're.
So maybe, like, you're talking about it being murky, right? Like, is there any thought about what exactly the mechanism is here? Like, what are there specific, bugs that are the ones they're interested in? And follow up,
question. If they know those, is there any talk about like just like isolating those specific days and like centralizing that? So
The good question. And so first, like nobody knows exactly what's in the stool. I mean, it's like people like we try culture like traditional microbiome methods are to culture it and we can grow, dozens, scores, hundreds of bacteria. But when you do more sophisticated stuff, like looking for amplifying looking for bacterial DNA and amplifying it, it turns out there's thousands of organisms, some of which have never been cultured, and we may not be able to culture, but they are in there. Which ones of those are relevant? Hard to know. And it's, when you're looking at, okay, which of these 6,000 organisms should we be focusing on? That's tough to do, especially if you can't grow some. But people have tried, that's the whole point of probiotics, lactobacillus, bifidobacterium. There's all sort of like, I confess that I have a bunch of favorite podcasts I listen to, and a lot of them are sponsored by, various, gut health things. And it's all grift. I mean, there's none of these like have any good data for them whatsoever. But, as I sent my resign email to the government, I mean, the obvious next place for me to go is just to design a gut health probiotic, and people buy it.
Yeah, they get a celebrity involved.
I should go.
George Clooney's tequila, but George Clooney's stool.
Oh, that's a fact.
It has just launched a probiotic vitamin supplement. So
Yeah, I mean, this is the thing to do. It is just astounding. AG1, I think, is one that I keep hearing about.
I am 8.
Yeah, it's painful. No, so we don't know. And when people have studied actual probiotics or stuff, very rarely is it like good trials don't show anything.
Can you, I don't say it and they won't get a vaccine.
Oh, Chris, you're hurting me.
Can you, is your study was done before there was a toxin?
There was the toxin test, but then our definitive test was toxin test if they sent the sample in. So it was, and there was no even amongst those that had the toxic test that confirmed there was no difference. That was obviously a smaller share.
One more question, Alan. Quick. Right at 10 to dot 5.
Just real quick. So I work in the area of human milk, and milk donation is something that happens freely all around the country. Yes, there's some control, but we don't know what's in milk either. Every woman is different. There's going to be lots of different things, and yet it's classified as a food, and therefore it negates all this stuff. But we still don't know what's all in there.
Yeah.
We completely do not.
Yeah,
And we do not know why milk has its effects on the baby, future baby. So this kind of makes me think, like, if we could re-engineer this historically, I mean, could this have been a food?
You got some marketing problems there. But yeah, I mean that was like, what is a drug? What is? I mean, I've had the same, very completely different topic, but there's a new HIV preventative medication that's injectable, and it's hugely effective. Like in the trial, it was 100% effective, and you give it by injection twice a year. And we clearly think of it as a drug. This is not a vaccine. That it's not stimulating your immune system. But from a patient standpoint, it sure seems like a vaccine. It's like, wait a minute, like you tell me to get 2 COVID shots a year. I can't tell the difference between the 2 COVID shots a year versus the 2 HIV shots, other than the 2 HIV shots are 100% effective, and the COVID ones are good, definitely not 100%. So it's like, yeah, what do you call it or classify it? And clearly the HIV is a drug, not a vaccine, but we're truly well. And so if we don't call this a dietary supplement or, yeah. So that was sure.
Well, we are at the top of the hour. Thank you so much, Dimitri. That was absolutely fun. That's now time for you to say thank you for coming to the WBOM and doing at least some of the merits at WBOM class. Souvenir. And for everyone else, we have a PhD student class that's starting here in 15 minutes. So most of you have started to slowly make your way out, and PhD students can stick around. Thank you. See you in 2 weeks. This is also